is the formations of nucleosome core particle and nucleosome filament. The nucleosome is known as bead on string. Each bead is a nucleosome, the basic units of DNA packing. There are two major groups of protein in chromatin, which is histone and non-histone. Histone is well-defined basic protein, while non-histone is mainly widely used. Histone is a small protein molecule, which is contain more than 100 amino acids. They have a positively charged. Some histone function as spool for the thread-like DNA to wrap around. This is the figure of nucleosome core particle.
Let's proceed with the formation of chromatin fiber. Chromatin is a mass of genetic material composed of DNA and proteins that condense to form chromosomes during eukaryotic cells division. The primary function of chromatin is to compress the DNA into a compact unit that will be less voluminous and can fit within the nucleus. Chromatin consists of complexes of small proteins known as histone and DNA. Histone help to organize DNA into structure called nucleosome by providing a base on which the DNA can be wrapped around. A nucleosome consists of DNA sequence that wrap around a set of 8 histones called an octamer, as shown in the figures. The nucleosome is further folded to produce a chromatin fibers. Chromatin fibers are coiled and condensed to form a chromosome. Chromatin makes it possible for a number of cell processes to occur, including DNA replication, transcription, DNA repair, genetic recombination, and cell divisions. The figure shows the chromatin fibers.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes known as chromatin. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the 2 groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of 2 genetically identical cells. To summarize the entire cycle, G1, S, and G2 phases occur during interphase while the cell is growing and preparing to divide. Mitosis, or the division of the nucleus, consists of 4 phases: prophase, metaphase, anaphase, and telophase, and in these phases duplicate copies of the chromosomes are split equally. Finally, the cell's cytoplasm divides during cytokinesis and results in the formation of 2 genetically identical daughter cells.